My name is ElenaSchroeter. I'm a 2017 Arnold O. Beckman postdoctoral fellow, and I'm a molecular paleontologist.
Whereas traditional paleontology uses the shape of bones of extinct animals to investigate their biology, physiology, and evolutionary relationships, molecular paleontology seeks to explore these topics using biomolecules preserved in their fossil tissue.
My research focuses on detecting proteins in fossilized tissue by using mass spectrometry to identify their sequences. My fellowship project sought to analyze 5 different specimens of Tyrannosaurus rex for preserved proteins using mass spectrometry. The core of this project was to develop a protein extraction methodology that would allow me to overcome the obstacles associated with analyzing fossils by this method.
Analyzing fossils in mass spectrometry has inherent challenges that aren't present when you analyze fresh bones of animals that were alive not so long ago. This is because decay products present in the fossil can interfere with the mass spectrometer. When you extract proteins from recent bones, the solution that comes out is typically clear. When you extract proteins from fossils, however, the solution can be dark and cloudy, full of decay products called humics. Because humics behave like proteins in solution, they are very hard to separate from the proteins we wish to analyze. However, if you leave them in the sample, it can create a ton of problems for the instrument, including clogging chromatography tubing, breaking your spray tips, and suppressing the electrical charges that allow the instrument to detect and measure any biomolecules that are present. In the first year of my project, I developed a workflow that was optimized to remove these interfering substances while simultaneously concentrating proteins extracted from multiple pieces of bone.
It was hard. I tried a bunch of different sample cleanup methods from the published literature, and some of them were all right, and some of them were just awful for fossils. In the end, I ended up combining a couple of different ones into a workflow that removes humics at multiple stages of the process.
I then tested my workflow on fossils from a recently extinct bird called a moa. Even though these fossils were only 1,000 years old, they were very dark with humic substances. Previous analysis of these fossils had shown they contained collagen 1, 2, and 5.
So I knew these fossils had both a ton of humics and some preserved proteins. So I knew it would be perfect for testing my workflow to see if I could remove one without losing the other.
When I applied my method to these fossils, my samples went from very dark concentrated protein extracts to clear samples that were virtually identical to the blank controls.
The first time I saw the final samples, I thought to myself, there's no way there's still protein in these samples. I'd never seen fossil extracts so clean, and I thought for sure my method had just removed everything, including the proteins.
But when I ran the fossil samples on the mass spectrometer, not only did I identify collagens 1 and 5, but I identified 16 additional proteins that hadn't been found in the original analysis, and which had never been seen before for this animal.
Even though my method did exactly what I had hoped that it would do, I was a little shocked. Pleased, but shocked.
Now that I knew my method could both concentrate and clean proteins from fossils bearing a lot of these decay products, it was time to apply it to my Tyrannosaurus rex fossils. I applied my workflow to 6 different specimens of Tyrannosaurus rex from different localities, as well as 2 sediment samples and 3 buffer blank controls.
It was a lot of tubes.
All of them were significantly clearer in the end than when they started, which was a great sign, though some of them seemed to still bear a small amount of humics even after all the filtering.
This is interesting because it points to something that's happening geochemically with the ancient fossils that wasn't happening in the 1,000-year-old bird. Whether or not this is due to the differences in ages or burial environment is still a question that needs to be explored.
Unfortunately, initial mass spectrometry analyses of these samples have so far only identified common lab contaminants that we expect to find in all samples, even the negative controls. So far, no bone proteins. Even though our analysis didn't find any bone proteins in these samples, the fact that we found things like human keratin and trypsin is actually good news. Typically, in fossils like these that start out with dark humic substances, the decay products actually prevent the machine from imparting the electrical charge that allows it to detect molecules. As a result, In fossil samples, we typically don't see any proteins at all, even the ones we know are present, like the trypsin that we put in as part of the experiment.
The fact that the machine can see these proteins means that enough humics have been removed from these samples for the instrument to work properly. And even though the goal is to get bone proteins, this is still a big step in the right direction, one obstacle down.
The next step for this research is to apply this method to more freshly excavated fossils, as opposed to ones that have been in storage for years.
This past summer, we dug up the skeleton of a new species of mosasaur that even has preserved pieces of cartilage. I can't wait to grind pieces of it up and see what this method can really do. The power and lasting impact of Dr. Beckman's contributions to science cannot be overstated. His technological innovations gave all scientists everywhere the tools to answer a wide variety of questions, and that continues to this day.
In fact, the centrifuge in my lab is a Beckman.
Sometimes I wonder if he could have envisioned the technology he developed being used to study dinosaur biomolecules. Maybe, since the technology he developed has been used to study practically everything else. Learning about his incredible legacy has changed how I think about my research, in the sense that we should always think beyond our own questions and always think about how we can use our skills to serve the greater good.
As a new research assistant professor at North Carolina State University, I am part of a new NSF project that will evaluate the protein content of fossils preserved in carbonate concretions from various times in Earth's history when the atmospheric carbon dioxide was much greater than it is now. It is our team's hope that elucidating the microbial processes that sequestered carbon to produce these fossil-bearing concretions will shed light on how microbes might be harnessed to help address global climate change.
So the methodology that I developed during the course of this fellowship will be directly used in research that investigates ways to build a sustainable future on this planet for everyone who lives here. I like to think that that's in keeping with Dr. Beckman's philosophy.